Welcome to The Explainer. Today, we're diving into a really fascinating randomized clinical trial. It's about a novel treatment for Alzheimer's disease, and it was published in JAMA Network Open.
You know, the study we're looking at today asks a question that's on a lot of people's minds. Can we use highly targeted, personalized brain stimulation to actually improve thinking and daily function for people in the early stages of Alzheimer's? All right, so here's our game plan for breaking this down. First, we'll cover the background and what the study was trying to do. Then, we'll get into the nitty-gritty of the design and methods. After that, we'll unpack the really key results, both clinical and from brain imaging. And finally, we'll wrap it all up with the conclusions and limitations. Okay, first things first. Let's set the stage a bit and really understand the why behind this whole approach.
So the problem here is pretty well known in the field. In early Alzheimer's, the hippocampus, which is like the brain's memory headquarters, starts to shrink. Now this causes a kind of disconnection from other key brain areas, especially the parietal cortex. The goal of this study, then, was to see if a 4-week course of personalized magnetic stimulation called rTMS could help repair that network and hopefully improve both thinking and that brain connectivity.
So how in the world did the researchers actually test this pretty ambitious idea? Let's take a look at the design and methods of the trial.
And the design really is the gold standard for this kind of research. It was a single-center, randomized, sham-controlled, and masked trial. What that means is patients were randomly put into either the real treatment group or a placebo, or sham group. And crucially, neither the patients nor the people evaluating them knew who was in which group. 30 participants, all confirmed to have early Alzheimer's, made it through the whole 8-week study. 18 got the real rTMS, and 12 got the sham stimulation.
Now, the intervention itself was incredibly precise. It really came down to 3 key steps. First, to select the target, they used each patient's own fMRI scan to find the exact spot in their brain that was connected to their hippocampus. Second, for delivery, they created a personalized 3D printed frame for every single participant. This locked the machine onto that exact spot every single time. Talk about precision. And finally, the protocol was 20 sessions over 4 weeks, delivering 1,600 pulses a day.
Okay, now for the part we've all been waiting for. What were the clinical results? Let's dig into what this study actually found.
Let's start with the main outcome. This chart shows a standard cognitive test for Alzheimer's trials, where a lower score actually means better performance. And you can see a clear split between the two groups over time. The rTMS group, that lower line, shows a steady improvement, while the sham group, sadly, continues to decline. And this difference wasn't just a fluke. It was statistically significant at week 4, and even more so by week 8.
And what's really great is that this cognitive benefit seemed to translate into real-world functional improvements. On one scale that measures clinical deterioration, the rTMS group held stable, while the sham group got worse. And on another scale that looks at daily living activities, the rTMS group showed a significant improvement by week 8. You know, this delayed effect is interesting. It suggests the treatment might be causing longer-term, positive changes in the brain. So you might be wondering, where exactly was this cognitive benefit coming from? Well, when the researchers broke down the scores, they found the effect was driven almost entirely by a huge, significant improvement in the memory domain. And that makes perfect sense, right? The whole intervention was designed specifically to target the brain's memory network. Now, in the interest of good science, it's also really important to talk about what didn't change. There were no significant differences on several other cognitive tests, like the MMSE or tests of frontal executive function. And this actually helps to highlight the specificity of the treatment, suggesting it's not just some general, non-specific cognitive boost.
So, the clinical results look pretty good. But were these improvements matched by actual measurable changes inside the brain? Let's take a look at the neuroimaging findings. And the answer is a clear yes. Some of the patients got fMRI scans before and after the treatment, and the analysis showed that the real rTMS treatment significantly boosted what's called functional connectivity, basically the strength of the communication line between the hippocampus and another area called the precuneus. You can think of the precuneus as a critical hub for memory, which makes restoring this connection really, really meaningful. And here is the aha moment where everything clicks into place. The researchers found a direct link. The more a patient's brain connectivity improved, the better their cognitive scores got. This is powerful stuff. It provides strong evidence that the patients weren't just feeling better, their brains were physically changing in a way that directly led to the clinical improvements.
Okay, so let's zoom out. What's the big picture here? Let's put all these findings together and look at the study's final conclusions, and, just as importantly, its limitations. The authors think there are 3 things that might be happening at a biological level. First, the stimulation could be helping something called long-term potentiation, or LTP. It's a fancy term, but it's basically the process our brain uses to strengthen connections between neurons. It's the foundation of learning and memory. Second, those delayed benefits we saw line up with how long we know it takes for the brain to rewire itself. And third, the fMRI changes just confirm that there was a real restoration of that key memory network.
Of course, no single study has all the answers. The authors are very upfront about the limitations here. We're talking about a pretty small sample size, some unequal dropout between the groups, and a sham design that couldn't perfectly copy every sensation of the real thing. They also didn't have data on tau pathology. These are all really important things to keep in mind for bigger, future trials.
But the take-home message here is still incredibly powerful and clear. This trial shows that a 4-week course of this personalized, MRI-guided brain stimulation leads to significant improvements in both cognition and daily function for people with early Alzheimer's. When you combine that with the brain imaging evidence showing real, plastic changes, these findings suggest that personalized rTMS could be a very viable, non-drug add-on treatment for this devastating condition.